you see the difference in how these triangles are labeled. That particular functional group is oriented very slightly differently between these two compounds. That tiny difference is enough to make a huge difference, such that our drug, KNS, does not promote all that dopaminergic activity, but it does protect cells equally to the degree that Mirapex does. Now, up on top, that's the difference between these two particular enantiomers. On top, we see the all too familiar Rilutec, which also bears very striking structural similarity on the left side of the molecule. But here's a very significant difference. This particular group may be what accounts for the principal activity of Rilutec, which is to modulate ion channels and excitatory amino acid release, glutamate toxicity. Now, our scientists know a lot about Rilutec. They've worked on it in their past lives in the pharmaceutical industry, and we're certainly very interested in it now because of the very close structural similarity to KNS. And our scientists look at this structure, look at the clinical data for Rilutec, look at the animal data for Rilutec, and have formed a hypothesis that that glutamatergic activity that we think provides the benefit from Rilutec may actually not be helping patients. That the advantage of Rilutec actually resides in that blue box, in the same structure that our molecule shares, and that the activity to block excitatory amino acid release may actually make patients feel somewhat weaker, which raises the, or addresses the rather interesting paradox associated with Rilutec, that while there is a survival benefit, and however modest in the placebo-controlled trials, it is significant, nevertheless, there's been very little functional benefit demonstrated in patients. Patients feel a little punk, some of them do, taking Rilutec. There's elevated liver enzyme involvement in some patients. So we are pursuing the development of the structure in the middle, guided in part by the theory that we have the half of the Rilutec molecule that may be working, but we lack the half that may be causing side effects and may in fact be inhibiting functional benefit. Now, I'm not going to try to present the range of data that we've developed in our lab and in our collaboration with Yale University to try to figure out how the drug works, but I'll show you one or two samples. On the panel on the left shows in the first group of 4 bars control cells. These SY5Y cells are cells typically used in experiments of this type. And we add the drug in varying concentrations, 0, 2, 10, and 20 micromolar. And you can see, just for purposes of control, that there's essentially no effect, although in the high dose group, normal cells just sitting there in the culture dish actually are a little bit happier in the presence of the drug. Now, that could be artifactual, although the finding is statistically significant, and we're not out to treat normal cells just sitting there in a petri dish. On the right, where you see MPP, this is a toxin that has been added to the cells. It's a standard toxin used to determine what may protect cells from various insults. MPP plus produces a Parkinson's-like phenotype. So as we add this particular concentration of MPP to the cells, you can see in the red bar where there is 0 KNS, there's a substantial reduction in cellular health. As you increase the concentrations of KNS, you begin to recover and ultimately exceed, to a statistically significant degree, the health or the vitality of these particular cells. We're carrying on this work now in neurons, and we'll initiate it as well in motor neurons. Now, back to Mirapex. You can dose a cell as high as you want, perhaps, without necessarily incurring a clinical side effect. So people have experimented with the neuroprotective properties of Mirapex. In this particular case, a group in the UK in liver, cells derived from liver. These scratchy little images on the left are electrophysiology recordings, patch clamp recordings, from the edge of the particular liver cell mitochondrial component to measure what's going in and out of that particular organelle. When pramipexole is added, the electrical, the ion channel activity from these mitochondrial components taken from liver changes extremely abruptly. And I can show you in our own experiments, this is from our collaboration with Yale, that in the top panel you see the control where from the outer mitochondrial membrane a channel is open. In the second panel, when you add our drug, the channel closes very quickly and remains closed. And in the bottom panel where it says wash, we actually remove the drug and you can see that the channel is back open. Now, what is this particular channel? Our scientists are focused on an area of research that's been underway for several years on a particular cellular phenomenon known as mitochondrial permeability transition. This activity involves the opening of a pore in the mitochondrial membrane. And among things that traffic within that pore is a chemical called cytochrome C. As cytochrome C is released through the pore from the mitochondrion, it initiates the process known as apoptosis. We're now experimenting to determine to what degree our drug inhibits the release of cytochrome C, inhibits the apoptotic process, and in fact inhibits the formation of this pore. And we have data that will be presented at the neuroscience meeting in the next 2 or 3 weeks that shows a dose-dependent effect on each of these activities. So we know it works in a petri dish. Does it work in people? There have been many drugs that have worked in petri dishes and not in people. There have been many drugs that worked in the SOD1 mouse and have not worked in people. Our drug has been shown to extend survival significantly in the SOD1 mouse. But so have drugs that have gone on to make no difference in ALS patients, and in fact, in a few cases, to make ALS patients worse. People aren't mice, even though it's all we have to go on in terms of animal modeling. So we are now taking our drug forward in clinical studies. One of the findings that we made in animals that was significant, however, is that our drug gets into the brain. One of the big challenges, and a scientist like Dr. Piora would, I think, attest to this, in developing drugs for neurological or neurodegenerative conditions, is actually getting the drug through the blood-brain barrier to the site of action where it can make a difference, in the central nervous system. It's very difficult to get a foreign object through that blood-brain barrier. Our studies in a variety of species have determined, mammalian species have determined that our drug concentrates in the brain preferentially, that however much is circulating in your bloodstream, the ratio that is present in the brain is as little as 6 and as much as 15-fold higher. So we know we get the drug to the brain. That's a big part of it. Another important criterion is how safe is it, particularly at the sort of heroic doses that we're contemplating administering to ALS patients. So we initiated a series of phase 1 studies. These were conducted in healthy volunteer male and female patients. There were 3 studies in total. The total number of people that participated in the studies was 82, of which approximately 75% received drug and the remainder received placebo.